I'm going to be reviewing enzyme classifications. There are 6 types, and the way I like to remember them is by using the following mnemonic. O, how, Lisa likes to isolate herself. And notice how I misspell the word likes so that it looks similar to the enzyme. So with oxidoreductase, these enzymes catalyze reactions that oxidize or reduce. There can be a proton exchange, an oxygen exchange, or a transfer of electrons. Here are a few types. So we have oxidase, which are enzymes that catalyze reactions where oxygen is the final electron acceptor. Reductase, or enzymes that catalyze a reduction reaction. Dehydrogenase, which are enzymes that catalyze reactions that remove hydrogen. They transfer one or more hydrides to an electron acceptor, such as NAD+. And so the reaction can look similar to this, where electrons are transferred between B and A. Hydrolase reactions, or enzymes, use water to break a compound into 2 molecules. A few types of these enzymes are phosphatase, which cleaves phosphate, peptidase, which cleaves peptide bonds formed between amino acids, nucleases, which cleave phosphodiester bonds formed between nucleotides, lipases, which cleave lipids such as triglycerides, amylase, which cleaves starch and glycogen into sugar found in the saliva and pancreas, pepsin, which cleaves proteins into polypeptides. This digestive enzyme is found in the stomach, and so this enzyme can look similar to this. Ligase enzymes, also known as synthetase, catalyze reactions that join substrates through addition or synthesis reactions. Condensation, also known as dehydration, may occur, and ATP is oftentimes needed. This enzyme usually occurs with larger molecules. So the ligase looks something similar to this. Lyase enzymes catalyze the breaking down of chemical bonds without the use of water. This produces 2 products. A unique characteristic of this enzyme is that it can catalyze the reverse reaction of its mechanism. For example, it is able to combine 2 usually small molecules in one. When this happens, it is known as synthase. And so lyase can look similar to this type of reaction. Transferase enzymes catalyze reactions that move functional groups. Kinases are an example of this type of enzyme. Kinases are able to transfer phosphate groups, usually from ATP, since it has 3 phosphate groups, to another molecule. And so it can look something similar to this, where X is a functional group. Isomerase enzymes catalyze reactions that form isomers of a molecule. Fumarase is an example of this enzyme, as well as phosphohexose isomerase, also known as PHI, which is used in glycolysis. And so the reaction can look something like this.